Two peptides can share the exact same amino acid composition, the exact same molecular formula, the exact same intact mass on the mass spec readout, and still be completely different molecules. Scramble the sequence, swap a stereochemistry, and the number on the readout doesn't move, but the biology does. So if the intact mass can't catch that, what does? Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams to prove fragment by fragment that the sequence on a certificate of analysis is the sequence actually in the vial. Today we are breaking down peptide mapping, the test regulators lean on hardest to confirm identity and why intact mass alone was never enough in the submissions to the Health Canada and FDA. Peptide mapping wasn't always the industry default. It earned that position. In the late 1990s, ICHQ-6b formalized peptide mapping as the standard identity test for biotechnological product. Through the 2010s, the FDA's synthetic peptide guidances extended that same expectation to chemically made peptides seeking generic approval. Then the GLP-1 boom hit and scrutiny on sequence confirmation intensified. Today, a peptide dossier without a peptide mapping study isn't considered a complete identity package by any regulator for any peptide. So what peptide mapping is? You break the peptide into small, predictable fragments with a specific enzyme, then run them through liquid chromatography and mass spectrometry to record each fragment's mass and order. Digest a reference standard the same way and lay the two fingerprints side by side, every fragment matching in mass and retention time, not just the total lining up. One map confirms the sequence itself, disulfide connectivity where it exists, and the exit residue carrying a modification, and any fragment that doesn't belong. Here is the fact that surprises people outside the lab. Two peptides can share the exact same intact mass and still not be the same molecule. Scramble the order of a few residues and total mass often doesn't change at all. Same atoms, wrong molecule entirely. Swap an L amino acid for its D mirror image and the mass shift is essentially zero, even though the molecule now behaves differently in the body. Intact mass confirms weight. It says nothing about order, nothing about stereochemistry. Peptide mapping is the routine test built specifically to read order, which is exactly the gap a same mass imposter hides in. Why does regulatory review lean so hard on this one test? Three reasons. Patient safety, a sequence error or the wrong stereochemistry can change potency or trigger an immune response nobody predicted. Approval risk, incomplete sequence coverage is one of the fastest routes to an identity deficiency later. And life cycle control, a new raw material lot or synthesis route calls for the map to be run again, not assumed to still hold. If you have ever watched a submission stall over a single unmatched fragment, drop a comment. It tells us exactly what to dig into the next. Not every peptide is equally easy to map. Cyclic, lipidated, and disulfide-rich peptides sit at the top of the difficulty pyramid. A fatty acid tail or a locked ring can block the enzyme's usual cut site. Modified linear peptides sit in the middle, and acylation or pegylation shifts a fragment's mass enough that you need to know where to look. Simple linear peptides sit at the bottom, clean digestion, straightforward coverage. The more structure a peptide carries, the more your mapping strategy has to earn that coverage rather than assume it. Three patterns keep showing up wherever sequence confirmation decides a peptide's fate. In synthetic peptide ANDAs, a blocked cleavage site that digestion never resolved is a recurring cause of identity deficiency letters from FDA. In compounded and unapproved GLP-1 products, some material reaching patients was never mapped against a reference standard at all. And in complex peptide and biosimilar programs, disulfide mispairing and truncated variants have slipped past intact mass, caught only once a proper digest was run. At Resolve Mass, we have supported sponsors through exactly this kind of investigation. One fragment that wouldn't match the reference digest, traced to a missed cleavage site and fixed with a rebuilt digestion method. The peptides look different every time. The failure point is almost always the same fragment nobody accounted for. So, how does a lab actually build one of these maps? 5 steps. Digest. Choose an enzyme, sometimes more than one, that cuts the peptide into predictable fragments. Separate. Run a digest on liquid chromatography so every fragment elutes at its own retention time. Detect. Mass spectrometry records the exact mass of each fragment. Assign. Match each observed mass back to the expected sequence. Compare. Overlay that map against a reference standard digested identically in the same conditions. Skip any step or use an enzyme that can't reach a lipidated region. You are left with unconfirmed sequence, not a minor gap. So, what should you walk away with today's discussion on peptide mapping? Peptide mapping confirms sequence order, not just the molecular weight, and order is where imposter molecules hide. Complexity should shape your digestion strategy from the start, not after a gap reappears. Coverage gaps, not dramatic errors, are where identity claims usually fail, and a map runs once can't be assumed to still hold. If you're developing a peptide therapeutic and you need a CRO or CDMO partner who treats sequence confirmation, peptide characterization as a core science, Resolve Mass Laboratories supports pharma and biotech teams from first digestion method development all the way through submission to FDA, Health Canada, and European EMA submissions. If this made peptide mapping click to you, please hit like and subscribe this video channel so it helps us to create more and more this kind of informative videos. Drop a comment if you are working on these areas. Thank you so much for watching this session and see you in the next one. Until then, goodbye for now.